Yeah. Perfect.
I need to get a program one time. She could. I have the same things.
Yeah, that's true. Yeah. Everybody. 20 minutes. I think there's yeah. Somebody like, okay, yeah. It's like,
Yeah.
I was like,
All right.
yeah.
All right, we ready? All right, well, welcome to the seminar today. And it's really just sort of like it's axiomatic, but it really is a pleasure and I won't say an honor, but it's to introduce someone I have deep respect for and have known for many, many years. So when you say it's an honor, that's like introducing a pope or something else. So Steve Mackesy is a professor in the biology department at University of Northern Colorado. He got his, just to do the official part, he got his bachelor's and master's at UC Santa Barbara. And I don't know if there's people online and stuff, his master's with a sort of a, well, I'll say famous or maybe / infamous by Sam Sweet, very, very colorful character there, herpetologist. He then did his PhD at Washington State. He worked with a fellow named Ken Cardong. One of my former students worked with him too. And Ken is not with us anymore. It actually went too early in my opinion, but at any rate, really very interesting comparative anatomist, and probably still has got the textbook, you know, the years after he died. And then so Steve will be talking about some of that, but then he his kind of shift, kind of like I did too. He shifted to his postdoc at Colorado State University, where he worked with Tony too, and this is where he started doing the biochemistry of the venoms, which was a like a turn from a person doing comparative anatomy kind of thing. So again, something like I had done. Steve has well over 200 publications. He's published 2 books, Handbook of Venoms and Toxins of Reptiles and the Biology of Rattlesnakes, the second, like round 2, I guess. And Steve is somebody I would say is a really great example of a guy that does really good field work and really good lab work. So for you students who are like, well, I don't know, do I have to be like this or that? Like you can do both, and you can do both really well, but I'm not going to steal his thunder. I'd say it's probably his field, and so he maybe a little bit not his field, but his work maybe a little like mine too is not, it's not inaccurate to say he's practically unique in that. He may argue. Steve has had many postdocs, grad students, and undergrads, and he is well known in the field for being a super mentor. And those of you who met with him today have figured out why. And he's also won, which is nice to see, has won many research and teaching awards at his university. So you'll see that today again in his talk. And so he's really, you know, as I said, really known as a great mentor of all time. I've known him for years. We both couldn't figure out where we first met, but at any rate, it's a really great pleasure to have him. Yeah, exactly. Both of us to have Steve out here to visit and to talk about his great work, which is snake venoms, ecology, evolution, and omics. So Steve, take it away.
Well, thank you, Bob, and thanks for the invitation to come here and talk about something that's very near and dear to my heart, that is venoms. And it's been a pleasure meeting with a number of you today and talking about a wide variety of things and even some science as well, right? So what I'd like to do today is give you an overview of some of the different systems that we see among venomous snakes. I'm not really going to get into taxonomy and phylogeny per se, but just put it in that sort of a context. And then an overview on venom composition, what makes up venoms in reptiles. And so we'll spend a little bit of time of that just to put all of the later material into a context. From that point, then I'm going to present to you what I refer to as 3 vignettes. And these are discrete areas of some of the research that we've done over the years that include things like looking at this protein we've called a relocator protein that allows for very efficient tracking of prey and also has some kind of unusual applied properties to the same molecule. Look at some aspects of comparative genomics and how this means that if we look at a variety of different species of snakes, what kind of trends do we see in overall venom composition? And then one last thing, talk about some stuff that we started a long time ago and have developed even in the last, well, continuing into the present right now, of how snakes actually store venom for long periods of time. And it's rather problematic. And obviously they solved that problem a long time ago, but they didn't tell us how they did that. So we've been interested in trying to figure that out. Then just very briefly wrap things up and give you some take-home points. So if we think about snakes, there is a wide variety of there are a wide variety of snakes found globally. But there was an event that happened roughly 100 million years ago, a happy accident leading to the evolution of venoms and venom delivery systems in a particular group of snakes, advanced snakes, and in particular, the vipers and the elapids, vipers containing rattlesnakes and their allies, the elapids, cobras and their allies, have very, very potent venoms, and we see venoms characteristic in that. But there's another group that include 2 to several families, depending on whose cladogram you want to believe. And these are rear fang snakes, and they have historically been in the family Colubridae, and they way outnumber the front fang snakes. And so we're going to talk a little bit about all of these guys. Well, one of the reasons I got interested in snakes is just, these are cool, beautiful animals, right? They show all sorts of interesting morphologies. If we look at the vipers, we see a variety of different types of forms, old world, new world forms. The elapids, same kind of distributional patterns globally, but more tropical in their distributions. Some of them actually marine species, but most of them terrestrial. And then the family Colubridae, the rear fang snakes, a wide variety of different types, ranging all the way from very tiny species to moderately large species. Okay. You'll notice we're not going to talk about boas and pythons and things like that. They're kind of so they never had. Well, just for completeness, we do have a couple of truly venomous lizards, and these are in the family Helodromatidae, include the beaded lizards and Gila monsters. Fascinating animals in their own right. I'm not going to really talk about them other than just to mention that they have submandibular glands here associated with grooved lower teeth. It's a very different delivery system, very different derivation of the tissues, but it does contain a lot of the same protein families as we see in cobras and rattlesnakes venoms. So it's an interesting and enigmatic system here because they feed on things like eggs and nestlings, rodents and birds, neither of which put up much of a defense, right? But the adults might. And so it probably is largely a venom that has evolved for defensive purposes. With the snakes, it's primarily a trophic adaptation that facilitates feeding. Okay. Among the rear fang snakes, we see a variety of different morphologies. This is one we worked with extensively. It's referred to as an Amazon puffing snake. It gets about 7 feet long. And it has on the maxillary bone some enlarged rear teeth in a series here. There are up to 3 that make up this delivery system. If you look at the venom gland itself, it's a Dubernau's venom gland. It has distinctive features. One of the most important of those is it doesn't have an adductor muscle associated with compressing the gland. Instead, they actually sit there and kind of bite down hard. And I think what's happening is they're compressing the gland passively by bulging the adductor muscles on there. There's nothing specialized that actually impinges on the gland. Front fang snakes, including the elapids, have a very different sort of system than the colubrids. What we see is a large venom gland here. There is often an accessory gland then leading to the base of a small fang. It's a so-called fixed fang. The maxilla can move a little bit, but it can't rotate it. And so it is something that's delivered fairly superficially into wounds. The one we've worked with the most is in the rattlesnakes, which are an example of vipers and pit vipers. And this is a very large venom gland. And it does have an adductor muscle associated with it. Compressor glandulae is a differentiated part of the adductor that compresses it, so it can forcibly compress and deliver the venom very, very rapidly through a curved fang with a very nice orifice. It's very much like a hypodermic needle, right? So it has a long fang that can go deep into tissues. It does the whole business in about 1/4 to 1/2 of a second. So you didn't see Steve Irwin playing with rattlesnakes like he did with the lapis snakes. The other part of that is there's a very large space for storage in the gland. And so all of these finger-like projections here are glandular epithelial cells. And then the space between the ductules and the basal lumen is a very large space for storing a large volume. By large volume, I mean a 3 or 4 foot rattlesnake can produce about 1 milliliter to 2 milliliters of venom from a single extraction. So quite a large volume there. Okay. Well, the first thing we do is extract the venom from the snakes. And this is the starting point. And somebody I think asked how we do this. And it depends on the snake involved. And so if you have a rear fang snake like this boomslang or a lighter snake, we actually give them a 2-drug regimen, one to immobilize and the next to stimulate secretion. The others you basically can grab and squeeze and go from there. So something like a large rattlesnake or a large viper here produces a fairly large amount of venom as well. We've gone to anesthetizing the snake because I'm not getting any younger, I think. And it's much safer than what we used to do. And so it makes it safer for me and it makes it safer and easier for the snake. So, as Bob alluded to, looking at venoms and venomous snakes can involve a lot of different disciplines. And so as far as really getting into a number of these kind of studies really run the gamut across biology. So there's something in there for almost everybody. And so I've had the opportunity to work in all sorts of different sub areas of biology where we're using various tools that have come out of the various subdisciplines to really try to address particular types of questions, and oftentimes in concert using a variety of these to try to dissect or understand how snakes are doing things like keeping venom in their gland for 6 months at a time. How do you keep it patent and have it ready to go in a moment's notice? So, all right, so if we get to this is venom part of the talk. Venoms are primarily proteins and peptides. There's about 10% of other stuff. That is still very much open area of research and not much effort has been put into that area. But what we have here are not de novo poisons, but instead co-opted regulatory components of just normal systems. But if you put a regulatory component into a normally operating system in orders of magnitude higher concentration, all heck breaks out. It's not a good scenario. So they've done a lot with this. We have a lot of different, very, very potent biological activities in these venom components. If you look at all the different types of proteins that are possible out there, it's only a very small subset, about 32 to 35 protein families have been documented in various venoms, but a typical snake will have something like 10 or 12. And of those, only a few will actually predominate. What they have done though, is a particular protein scaffold works very, very well. So instead of going out there and grabbing something new, what we see is gene duplication. And once we duplicate all those copies of genes, then the opportunities for those being tailored and reworked into different activities has gone crazy. And that's the typical pattern and motif that we see in venomous snakes in general. Is we have these highly conserved structures that the structural features of the molecule all are highly conserved because that's what holds it together to form activity X, but everything else, including the functionality portions, is open to mutation. We'll come back to that in a little bit here. So basically what we have is combinatorial chemistry gone wild, and these have done quite a bit with that. So one of the things people recognized a long time ago is that they kind of flip-flopped the way that we see regulatory systems set up, and we actually have exons that are more variable than introns. And in most genes, we see the opposite kind of scenario. And basically that's just an expression of the fact that the molecules themselves are the same family of molecules, but the specifics have gone all over the place. Okay, so here's that list that you want to write down so you'll remember next time somebody asks. This is just some examples of the various families of proteins that are found in venoms in general. We can simplify that much by putting this in there. And by far and away, when we look across all the different venomous snakes, they fall into these four families primarily. Variable amounts of some of the other ones in various combinations. But in three-finger toxins, particular toxin family that we'll get to, phospholipases A2, metalloproteinases and serine proteinases are characteristic, certainly of vipers, and then some of these of the elapids, and we see them in various types. In our area of Colorado, there's another toxin, myotoxin A, that is a 4.8 kilodalton toxin, a little peptide that is only found in part of the population. And this is a very, very potent inhibitor of calcium pump regions in the sarcoplasmic reticulum. And snakes that have this can stop prey just like that. It's an absolutely amazing little toxin. Interestingly, it's very potent and very abundant in the northern populations and almost absent in the southern populations. Well, from a perspective of treating snake bite, this is kind of encouraging because up in the corner here, high toxicity, high abundance, that Chema Gutierrez put together a couple of years ago. These are the main characters we have to worry about neutralizing. Unfortunately, it doesn't fall out all that simply because if you neutralize one three-finger toxin, there may be two other homologs that are expressed in the venom that have very different epitopes on them. And so they haven't been touched by your antibody for that preparation. And the same thing with these other families. So on the one hand, simple, on the other hand, very complex in a different sort of fashion. Okay. Well, let's think about the evolution of toxins for a little bit. And primordially, or less derived scenario, is using mechanical means to hold on to prey. They're challenged, right? They don't have any arms or legs, and they don't have jaw structure and teeth that would allow for crushing or killing bites per se. Secondarily, with venoms, they do. But what we see, though, is in the venomous snakes, we have chemical-based predation. And depending on the lineage we're talking about, we may actually be using this remotely. So rattlesnakes and a lot of vipers are strike and release predators. They do the job quickly and then retreat back and wait for the prey to be incapacitated. Sometimes that's very, very rapid. But if we think of what's happening at the level of the gene, again, these are co-opted regulatory genes. And so these are normal compounds that are found in the body of the animal. They're duplicated and they're overexpressed in this very specialized region that we call the venom gland. Once they have gotten freed of the constraint of being whatever regulatory molecule they were for the snake, then all constraints are off. And what we see is that they go crazy. We undergo typically neofunctionalization. We can't have some that kind of go dead end lineages and subfunctionalized. But the name of the game also is then expansion of these families of proteins that are coded for by these specific genes. And so we get lots and lots of things that look superficially like they're the same basic molecules. So in addition, another selective force, of course, is the fact that things like mammalian prey or whatever prey they're feeding on are not static themselves. And so they have the capacity to evolve over time as well. And that seems to be the case as well. A recent paper that's still in press with a student looked at this in a system in Colorado where we have these two different sets of venoms, northern and southern phenotypes. And what we see is that there's very strong interaction here and the opportunity for co-evolutionary adjustments between both the predator and the prey. We're not necessarily claiming that's the case in particular, but what we do see is those populations that have myotoxin A, that small toxin I just mentioned, they really nail naive and co-localized prey very, very effectively. It's a very potent venom composition that the prey in their area has not yet caught up with. In the south, that particular concoction, if you will, is pretty well neutralized by some of these same species. And he's looked at this in 5 different rodent species. So it's kind of a neat little story that's developing along this line. Okay. Well, last real thing we want to focus in on here is what kind of systems are we targeting in the prey? And unfortunately, this includes larger animals like this that get in the way at times. And so peripheral nervous system and the neuromuscular junction in particular is a great place. All sorts of different animals that specialized toxins to hit a variety of target sites. Snakes generally go presynaptic or postsynaptic, associated with the receptor or with the release of the acetylcholine at the junction itself. There are toxins that hit specific muscular architecture. So there are some that actually cause damage to the myocytes themselves. And then like myotoxin A, there are calcium PUN inhibitors that allow for the muscle contract, but not to relax. And so when those prey are bitten, they undergo tetanic paralysis and they are immobilized, but not necessarily dead. And it's amazing how fast that can work at times. Vipers in particular, but really all of the venomous snakes have also gone up after the cardiovascular system. I'm not going to go through every one of these, but in fact, there are all sorts of different targets along this. And if you think about it, that makes perfect sense. If you have a vasodilator that is produced as a result of venom composition, vasodilation is going to lead to a crash of blood pressure, and that's going to help in that whole immobilization event. And that is a line that most of the rattlesnakes and a lot of the vipers have gone down. So we've got a number of different systems that are targeted by venomous snakes. And this is why a venomous snake bite is so challenging and dangerous, because it's not just one little system here or one over there, but all sorts of different systems simultaneously. So what I'd like to do now is just talk about some specific projects that we've worked on over the years. And this is one that has developed in various stages over several years as well. So if we think about chemically mediated predation, came across this black-tailed rattlesnake that had dispatched a chipmunk in the Chiricahua Mountains years ago, and very effectively took it out. And then when I came back down the canyon, it had swallowed it down. So we know that this works very well, and they are able to find their prey. Well, rattlesnakes and pit vipers generally use this type of sit and wait strategy. They're a strike and release predator, usually, unless it's very small prey. And so they're envenomating the prey and then letting it go. And the advantage to the snake is obvious. Rodents have very sharp teeth. Most of them don't like being killed. And so they will, in fact, fight back if they have any kind of chance, right? But it does create a problem. And that is they can actually wander away from where the snake struck them. Not all of them have myotoxin A. And so it can be a bit of an issue there. And they have to then relocate the prey. They use a process that's referred to as strike-induced chemosensory searching. This, once they bite something, it sets into mode the stereotypic behavioral pattern. And they then wander out, and they can very clearly differentiate envenomated versus non-envenomated trail. Well, this is an interesting scenario. So if you imagine a snake lying in wait, there's not a snake there. But imagine it's there and there's a mouse right there, and we have a strike. It's been envenomated, the mouse runs off. It wanders off in a variety of different directions, potential directions. Might go down a burrow, and as it does so, it's crossing maybe its own path that it's been using as it's foraging around doing its mousy thing. And there may be also conspecifics or congenerics or unrelated species leaving trails all over the place as well. In addition, we can have a scenario where they're doing this in the dark. So there's no visual cues at all, right? And so the basic question is, which track do you follow? How do you do this? So many years ago, a colleague, David Chazar down at CU Boulder, came up with this whole idea of strike-induced chemosensory searching and this idea of being able to recognize envenomated versus non-envenomated prey. And he showed with lots and lots of papers and lots of different species of snakes that This is kind of a very common theme seen in a variety of different species. We started collaborating with him on a number of different projects, and one of these was to determine this minimum perceptible dose of venom, which turned out for the Western Diamondback Coryllis atrox to be 7.1 mg of venom per mouse. Well, that doesn't sound like very much, and it's not much compared to what a big Western Diamondback can actually produce. But if you look at the toxicity towards a lab mouse, it's way, way over that level of toxicity, what it takes to kill the mouse, which is something on the order of 80 micrograms. So basic question on that was, well, why are we overdosing in this sort of fashion? So we came from that perspective of, yeah, they can discriminate, but it takes a moderately large amount of venom to do that. And so we had a working hypothesis on that one of these groups of proteins, the metalloproteinases, cause structural damage, hemorrhage, necrosis, all sorts of very nasty changes in the tissue, kind of a pre-digester or tenderizer, was probably the one responsible, just because that has very profound effects that happen very quickly. So first thing that we did is use size excluding chromatography to take the venom apart, look at large, medium, small molecules. And then instead of testing the whole venom and looking at prey discrimination ability, we use the fractions of those venoms. Okay. So he came up with a method where he allows the snake to then spend time over an envenomated or a non-envenomated carcass. Again, the important or an interesting aspect is this is with mice that were euthanized with CO2. So they're not even alive, and they can discriminate those. This is what he calls the tongue flick assay. I'll be happy to talk to you about that. We go through the venom and ask, where do we see all those different activities that we talked about? And so that's what the colored lines here are, the enzymatic activities, catalytic activities in the venom. We tried all of those. And then there was this one down here. Which one do you think they? This one that's in very low abundance here. Well, it turns out after a little bit more work with this, that is a very specific type of molecule that had been known in venoms called a disintegrant. And that has a very conserved structural feature to it as well. And we're able to purify it and then look at it, get a mass spec, and also get N-terminal sequencing. So we got a very exact match with databases here as far as what that molecule was. And so it's a kind of a curious deal. Now, I want to draw your attention to one aspect of the structure, and that is this RGD area here. And that's illustrated in the model right there. And just kind of file that away from them. So summarizing what we've done here, what we have is a molecule that's in the venom that in fact is responsible for this whole capability. We have this problem that's set up where once you let something go, it's going to run away. It actually might escape in 3 dimensions. That's more of a challenge, but keeping it in 2 dimensions, still trying to find it is challenging because that trail is crisscrossing chemical trails of God knows what. Interestingly enough, that mode of envenomation is seen only in vipers. And we've seen, we did this whole experiment twice because the first time through I didn't believe the results, that it's due to this molecule and all by itself. This can be introduced into dead prey tissue and it still causes some kind of a change that the snakes are able to pick up on. So their chemosensory perceptions are absolutely tremendous. It's also a very minor component. So this helps explain part of the reason why snakes get such a large dose, because they need to get it up high enough level where they actually activate the system so they can relocate. Also turns out that a lot of prey have resistant mechanisms, and so this helps to compensate for that as well. Lab mice are okay for a model, but they really miss a lot of the picture. Okay. The disintegrants are also found only in viper venoms, so we see the behavioral pattern, the ability, and the molecules that seem to be responsible for it, all encapsulated in the same group of animals. So it's kind of a cool deal. But other work has been done with disintegrants in a very different line of research that had nothing to do with how the snake is using this particular compound. So we started to look along that line as well. And this involves effects of toxin molecules and evaluating those on cancer cells in vitro rather. Sorry. So I had an undergraduate student that conducted a lot of experiments with a lot of different species of venoms. And in fact, not too surprisingly, when you give cancer cells exposure to venom, they don't like it, and most of them die. And some of them are very potently cytotoxic. You can see a couple species here with very different venoms that kill cancer cells very well. Well, that's okay, but they also do that to normal cells. They've evolved to digest and kill and mess up all sorts of normal systems as well. So it kind of seems not probably the best thing for a therapeutic, right? But this is a concoction of a whole bunch of different things. So if we start purifying disintegrants, we start seeing a different scenario. One is that when we look at cancer cells, they overexpress some specific markers, that is cell membrane components referred to as integrants. You remember this RGD motif here? What I like to explain these to students as is the integrants are kind of like the feet of the cell, and they're interacting with the matrix, they're interacting with other cells and other compounds, actually to move the cell along. And of course, cell motility when cancer cells become metastatic is when it becomes very, very problematic. So the cancer cells are using these as part of their whole motif for propagation and survival of cancer cells. So we asked the question, well, what can we do with these disintegrants? And I had a student that was very interested in this, Anthony Savioli, and did some fantastic work a number of years ago. And he did it with particular components that make up the extracellular matrix. And he's also done it with combination things like matrigel. But what he found is this compound, this disintegrant compound we called subconin after the snake that produced it, is not toxic to the cancer cells, but in fact, it does inhibit metastatic migration. And so it's actually doing this by binding to specific integrated components that are found on the cancer cell, or binding to the receptors of those that are found on the cancer cells, and then inhibiting that capacity for them to make that migratory movement on the matrix itself. Okay. He's used a variety of different techniques in cell culture and flow cytometry to show this, and some inhibiting of all these effects with specific antibodies. So we have a system here where it's kind of interesting because the snake actually is producing this compound as a prey relocator protein. But we have been able to repurpose this as a anti-metastatic or potential anti-cancer drug. A colleague working at USC has actually been working on this a lot longer, and they actually have had compounds in clinical trials. So it's just an example of where basic research can actually inform applied research and directions there. That we have this very unusual molecule that snakes didn't evolve this for us to use against cancer, right? Which sometimes people get that question. So anyhow, it's a very interesting and peculiar kind of compound. So, all right. So one of the things that's interested me for a long time is this whole aspect of these gene duplication events that lead to all of these different compounds with the same overall activity. And so we're interested in looking at the venom proteome of advanced snakes in a little bit more detail. And then asking the question, do we see patterns falling out, general patterns? The answer is that yes. This is kind of old school these days, but I still like running gels because they're very informative. They're very simple. I can have introductory students running them. They actually get publishable results in many cases. Plus, we know some of these compounds very well, so we can start seeing where they're present or absent in various venoms. And it's a very simple experiment. So we do this as a way of quickly canvassing variation, in this case, in the population of prairie rattlesnakes, and the COCs with the black bars are midget faded rattlesnakes. And we can see some pretty obvious differences between those species groups as well. So electrophoresis, still good. We've also, in my lab, had a graduate student, Cassie Modell, that developed very good transcriptomic techniques as well. Another student really went down the proteomics path. And between the two of them, the three of us, we actually have taken these venoms apart in a lot of different directions. And so we can use this multi-omic approach, and having a good transcriptome for the gland is very, very important for being able to do proper proteomics, because otherwise you don't have a database to compare it against that's very believable. So we do that, we can start asking questions about some of these little known snakes. This is a lyre snake from Costa Rica in a paper we published a couple of years ago on that venom. And what you see is that it's a fairly complex venom. This is HPLC traces here. We can then secondarily run on gels and then mass spec identify a number of these different protein families. And we see, like we've talked about before, there are a bunch of different components here. In this case, we actually have 3-finger toxins, 3-FTX in the corner there, in these rear fang snakes. So if we look at these broadly, we can start seeing patterns that are developing broadly across some of the family groups. So things like vipers have this type 1, type 2 dichotomy that we set up a long time ago. This is where we have venoms that either have very high levels of these digestive enzymes, catalytic enzymes that degrade structural tissues and things like that, or we have very low activity of those and very high levels of other types of activities, typically those that are compromising neuromuscular function in one fashion or another. Some of them actually actual presynaptic neurotoxins. And if we look at elapid snakes, they more are running down the line of type 2 type venoms, high toxicity, very low enzymatic activities and things like that. And if we ask what kind of patterns do we see in the rear fang snakes, only a handful, relatively speaking, have been investigated in detail, but we see the same kind of pattern of type 1, type 2. The metalloproteinases and neurotoxins, or the converse of that in the rear fang snakes. If you're a more visually oriented person, we can put that in this sort of format. Type 1 venoms that we see have lots of catalytic toxins in them. Type 2, in this category, have alpha or beta neurotoxins that are presynaptic neurotoxins based on a phospholipase scaffold. And we see that, and if you look at the sequence of these, and we've done this for a number of them, the ones that are found in Western Colorado are about 99% protein sequence identity with those that are found in South America. So very, very highly conserved structure on these molecules and in these animals. Okay. So we see this kind of pattern. If we look at elapids, we see a very different sort of pattern. They're putting all their eggs in a very different basket. In these, the hot colors here are the 3-finger toxins. Phospholipase-based toxins are shown in dark blue, and that is a general pattern that we see in the New World, Old World, beneath the sea snakes, very specialized elapid snakes. We ask the same kind of question of the rear fang snakes. Some go this way, some go that way. Makes a certain amount of sense because it's a very diverse lineage of advanced snakes. And so various lineages have really invested heavily in one type or another. So this is kind of an overview of what we see among the venomous snakes as far as the distribution of those various toxins. And those have different feeding and foraging strategies associated with them as well. Most of the elapids and the clubrids are a bite and hold type of predator, whereas the vipers typically bite and release. Okay. It's also provided us an avenue to start looking at some of the specific toxins. And I want to talk just a little bit about these 3-finger toxins that have a conserved molecular fold as illustrated here. This is actually superimposed 3 different molecules from 3 different species and 2 different families of venomous snakes. Their target is the same, the acetylcholine receptor and the alpha subunits they block, they bind to and block that receptor quite effectively. So the classic one is cobra toxin from many of the cobras of Southeast Asia. This picture was taken in a really suburb backyard. A student that fancied himself an importer had monocle cobras and 3-year-old brothers and sisters. Not a good mix, but made it through okay. If we look at how toxic they are to mammals, what we see is potently toxic, very, very low LD50 values. We've isolated some from this green vine snake, or rear-fang snake from Central and South America. It's non-toxic to mice. We've isolated from these mangrove snakes from Southeast Asia. It's non-toxic to mice. From brown tree snake from Guam, it's non-toxic to mice. We're starting to get into this monotonous, repetitive sort of scenario, right? Another species here, that puffing snake from South America, Amazon puffing snake, same kind of thing. It's non-toxic to mice. They're investing in producing this, and in some cases, this is a very major component in the venom. So, you know, what's the story here? Well, if we look at it not from a pharmacologist perspective, but from a biologist perspective, say, what are these things normally feeding on? They're probably not feeding on domestic inbred strains of lab mice, right? If we look at the same molecules and their effects towards geckos, hemidactylus geckos, in this case chicks, we did both lizards and birds, they're very, very lethally potent in these animals, and they cause the same kind of flaccid paralysis that we see with the cobra toxin. If we look at it with this toxin from the green vine snake, same kind of thing here. We looked at 2 different species, one a model species, the other a reasonably good mimic of a prey. They actually show a differential that's fairly significant between the toxicity towards the gecko, which is a model, and the anolis, which is an actual species of prey. Same kind of thing with this toxin from Amazon puffing snake. We've got the same kind of data with a couple other species, mangrove and the, I guess the mangrove is the only one we're dropping out here. But the Amazon puffing snake is an interesting one because it eats a wide variety of different animals. It feeds on mammals, feeds on lizards, feeds on birds. It also is not a constrictor at all. It'll do a body pin, but it doesn't constrict like that python I showed earlier on. They also have a separate toxin we call somotoxin. This one is lethal to mammals, not potently lethal, but still pretty good rattlesnake venom range, but it's non-toxic to lizards. So we've got this scenario here that we've only partially deconvoluted, and we have some ideas about what's giving rise to this specificity, but we really don't know for sure what it is. But basically, it's all wrapped up in that some aspects of the structure are very tightly constrained, and the non-structural portions have mutated all over the place. So just to summarize this kind of end of things, we can now catalog things at a level that we were never able to do, and we can go into a lot of different systems and very quickly, in some cases, get a good handle on what is in that venom from this species. There are some high throughput systems that are coming into play now, where we have a colleague in Denmark that thinks he can put through something like 1,000 venom samples a week. And so you have the opportunity then to dissect things at the populational level in kind of detail that were not possible in the past. We still see that only a few toxin families are actually dominant. And we see these kind of dichotomous relationships in the various families where we have this or that type of toxins are typically found in the venoms. But the important thing is that omics can only take you so far. And so there's some basic chemistry, biochemistry, protein chemistry sorts of things, include comparative toxicity assays and things like that towards natural prey or other types of prey that really can tell us a lot more about what's going on at the level of the biology of the snake. So we'd like to try to combine as many of these different tools as we possibly can. All right, the last thing I want to talk to you about has to do with this question of how snakes safely store this bioweapons armament that they have. And so we actually published a paper with bioweapons in the title. Long after I got a visit from military security. Not too related, though. But it is an interesting phenomenon. So if you think about the venom glands of snakes generally, but vipers in particular, because of the mixture of catalytic toxins, enzymes in there, they've got a bunch of different things in there. They store it in this gland for a long period of time, and in captivity, in a zoo setting where they're feeding dead prey, they may not use venom for years. But if you have a bite or if you have live prey in there and it's bitten, you can tell it's potently active and ready to go. So this is kind of curious because these animals actually contain in their venoms things that break up proteins. Well, the venoms are largely proteins. So how do you share proteins in these glands with proteolytic enzymes? So that's kind of the background that we had, and we were interested in dissecting that in more detail. As Bob mentioned, some of the work that I did as a PhD student was structural based and got into a lot of electron microscopy. It's been shown by Brazilian researchers that parasympathetic nervous system stimulation as a result of the release of pressure on the gland. When the gland's full, it's pressurized, or it's providing pressure on those systems. And when it's extracted, we lack that pressure. That is in fact enough to get gland production going, or venom production going. Cells within a couple of days max increase in height by about 100% almost. And what happens is they're forming huge amounts of RER, and they're basically turning into protein producing factories. This is then being packaged into vesicles. Those vesicles are going to be transported to the apex if they're not already there, released into the lumen, and then the cells return to a cuboidal or resting morphology. We have some secondary evidence though that there may be constitutive secretion going on, so low level secretion in the background, if you will. The ducts empty in this case, but at this point would be filled with venom, and we have a happy snake that's ready to go again. Its glands are completely full. If you take a snake, extract all of its venom, and ask how long does it take to replenish, it's about 2 weeks. On a more regional level, it's probably much less, and of course they have a large volume because they're a large glanded snake. In conjunction with Todd Casso's group in Texas, we've done a lot of collaborative work with them and have started to unravel some of the regulatory elements that are controlling this whole process. But basically, they've co-opted some of the kind of standard pathways for regulating protein secretion, and so a little bit more on that a little later. This also came out of some of that earlier work, and the main reason I have this in here is just to illustrate that for something like the snake venom metalloproteinases, SVMPs, that's just an example of how we have this gene duplication that has gone crazy, and there are bunches of them. And so in our prairie rattlesnakes, we have 11 or 12 variants of this same basic gene. These are derived from metalloproteinases that are part of a matrix metalloproteinases that are part of the normal system. We see that with the serine proteinases, the SVSPs, that interfere with blood coagulation and circulation generally, as well as the phospholipases and other toxins. This is Blair Perry in particular, an incredible kid, has in fact mapped all of these different genes onto the chromosomes. And so we know the chromosomal locations of most of these toxins. Myotoxin A was a real problem for them to map, has been found on chromosome 1 now. But most of them are on microchromosomes. And as far as a mechanism that provides for at least mentally easy evolutionary changes, it's kind of an interesting aside. But a long time ago, we were interested in this problem of storage and how they do that. And it turns out that there are compounds in the venom that are actually or potentially inhibiting. Some of these are peptides, some of them are citrate. Well, we looked at citrate, and citrate at the levels that occur in the venom, which is where that arrowhead at the bottom there is really not a very good inhibitor. Turns out there are also these peptides, tripeptides that are in the venom. They're kind of relative to something like a chelator that is a very potent inhibitor. These are low affinity inhibitors of the metalloproteinases. The metalloproteinases are where we focus because those are the ones that potentially could chop everybody else up. So we've got a system here where the presence of the metalloproteinases stabilizes incubation at 37 degrees over 100 hours, 95 hours total time, or in its absence, the enzyme falls apart. If you look at its distribution in vipers, North American vipers, South American vipers, we find it in both the venoms, and then we see those two HPLC peaks illustrating that. And if we look at the other clades, just illustrated by a cobra here and by a rear fang snake and false waters over here, what we see is that in those same places, we don't see evidence of those peptides. So we don't have that kind of a control system that's present in either of those. So it seems like something, again, limited to vipers. What was more interesting is this, and that is that when we did measurements on pH, the venom is actually stored at about 5.4 to 5.6, fairly moderately acidic pH. Talked to colleagues around the world, and snakes in North Africa, snakes in South America that they had measured pH were also at about that same level in the vipers. So that seems to be a common thing. Well, why is that of interest? If you look at bell-shaped curves of activity versus pH, we see a typical pattern. Most enzymes will have that kind of curve shape. If you ask where is it stored and where is it most active, this is where it's stored. And all of these are the tail of activity levels. Where is it most active? In slightly alkaline pH. What else has a slightly alkaline pH? Above neutrality. Now, this is one for the graduate students out there. When I was teaching a human physiology class, for which I had no preparation before, I was kind of annoyed at the whole premise. It just got kind of stuck on me. I was like, I had to teach this damn class. We got to gastric physiology, opened up the book and saw this cell type. And I was like, wait a minute, I've seen that before. And so that is shown here. And the most important thing here is that there's spaces in between these cells. These are secretory cells here. This is a cell that had been described a long time ago called mitochondria-rich cells. This is where you can data mine your own stuff from way back in the day. So we pulled up some of these electron micrographs and reinterpreted. It turns out that in the venom gland, we have a system that looks like this. In the gastric pit of the stomach, and to a large extent snakes, we see a very similar kind of morphology in the parietal cells. Okay. What are the parietal cells doing? They're producing massive amounts of HCl, acidifying your stomach. We have the same problem, and I asked my anatomy students this: how do you keep from digesting yourself when you're eating that hamburger? Because that hamburger looks a lot like you do. So think about that. So we had this morphological data that was very, very suggestive. I kind of skipping a piece in here where a graduate student did a lot of histochemistry and showed same kind of responses as far as histochemical staining between parietal cells in both the snakes and rat, and also in the morphology and the staining of the snake venom gland cells. Well, getting together with Todd's group again, we're looking at upregulation of genes in the first 24 hours in particular. And there's a huge number of them. And if you've ever done any of those kind of experiments where you're looking at upregulated, downregulated genes, it's kind of frustrating because it's like, well, we got 2,500, 4,000, 5,000 that are upregulated, 2,000 downregulated. How do you make sense of that? If you kind of limit that by using gene ontology terms, and this was pH reduction, that kind of condenses players in there a little bit without cherry picking data too much, right? Because I always feel like you're eliminating 99% of your data could have an effect. Well, the thing that lights up are these ATPases, and these ATPases are ion pumps. Right? So another student actually did some Western blotting. And the first thing we thought, well, the HK ATPase is found in parietal cells in large quantities. It's the one for translocating protons and acidifying the stomach. That should be our first look. And if you look at rat gastric membranes, rattlesnake gastric membranes, you see good signal here. And if you look at the venom gland, you see very good evidence for nothing at all. And so that didn't seem very productive. Fortunately, we had access to a set of antibodies to look at other ones. And so if you look at, it's kind of an ugly blot, but if you look at vacuolar ATPase is what you see is very, very strong staining in both of those gland tissues. And so that was definitely something that looked like it was promising. We were able then to follow that up with some microscopy and immunofluorescence. And what we see is these cells light up, and it's a limited number of them. And those are actually the mitochondria-rich cells. The other thing about them is they sit down in a pit. So when you do scanning EM, what you see is the venom surface, the glandular surface looks smooth with little holes in it here and here and here. And it's very reminiscent structurally of the gastric pit. A little more complex than the gastric pit, but same basic idea. So here we have those cells are lighting up for vacuolar ATPases. So what it seems to be is that we've got this scenario. We've got this armament of toxins that are stored in the gland. Some of them are autolytic, so they can break themselves down. I didn't mention it before, but they're about that far away from the brain on both sides of the head. So you've got this, one of my graduate students just defended me. He used the analogy of it's like strapping explosives to both sides of your head. Which it kind of is. But the snake has to protect itself from the effects of the venom, has to protect the venom's patency, but it needs to have it ready to go as an anti-predator deterrent and importantly as a way of getting food. So what we see are a couple of solutions, and these seem to be redundant solutions, and anything important in biology usually doesn't have a simple answer, and this doesn't either. So we see these low affinity inhibitors that are co-secreted, the tripeptide inhibitors I mentioned. We see that the venom is maintained at an acidic pH, and if you get into the chemistry of citrate, there are some pKa's, 3 different pKa's on citrate, and one of them is very close to that 5,4 range, so it's a good buffer in that region. And when we inject that bolus, then what's happening is we're going from an acid environment into a slightly alkaline, neutral to alkaline environment, and the low affinity inhibitors diffuse away. They're not bound tightly in there. They're high concentration but low affinity. They diffuse away. Goes into an environment that has 2 log steps, more friendly milieu, and they're ready to go and they do their thing. So Sam is the one that just defended on this, and he has taken the next step here in trying to go in and incapacitate those vacuolar ATPases. We have some specific drug inhibitors. And so if you've never tried to implant an osmotic pump in the head of a rattlesnake, I invite you to come by sometime. Actually, that didn't work at all because these are designed for rats, and it didn't seem like osmolality in reptiles was sufficient to drive the pumping. So we don't really know what happened, but we assume that's what happened. So wrapping things up here. Hopefully you'll agree with me. We've got a system, a series of systems that really is a pretty fascinating group of organisms and adaptations, and they're all different directions that we can go here. And the fact that these things are so variable means that we have questions to keep us busy for long periods of time, sorting out what's going on here. We know that all these different factors at some level or another influence this level of variation. But trying to tease apart, especially within a population or within a species, which are most important is challenging at best. So one of the cool things in this is that the phenotypic genotypic link is pretty tight, pretty obvious, and that's tied to fitness. If you have a venom that doesn't work, you're probably not going to get food very often. And so, in fact, lots of positive feedback on the directionality of that venom, but it may be more an example of stabilizing selection. So this is, a lot of people have started getting interested in venoms from this perspective in particular, plus the fact that we have all these omics tools that we can generate massive data sets very rapidly. So one of these is Todd Casto's lab down in Texas, and we have a continuing collaboration with him on a major project, really looking at how all of these different events in secretion and stabilization and storage are actually regulated. Stay tuned for that. It's pretty exciting. So The last point I want to make, though, is venoms really seem to be important for vipers. And in many parts of the world where we have vipers in an unmolested environment, they are by far and away the most abundant snakes and arguably the most abundant reptiles in those particular areas. So it serves them very well, and they've been doing very well. So with that, I'd like to acknowledge support from a number of different agencies over the years that have really helped to fund some of this work. And of course, most importantly are some of the many graduate students that I've had over the years, several of which have just been incredibly productive and are continuing to remain so. So we actually took this picture of the Everglades just before what was the last meeting that we would attend for another year and a half at the start of the COVID epidemic. We've also had opportunity to work with some outstanding colleagues. Todd Castro I mentioned before. Kinney in Singapore has been very, very helpful on a lot of different avenues. These two ladies here, also one in Singapore and one in Argentina. And then these two characters here who are PhDs in my lab, have graduated. Both are down at CU Anschutz Center and are heading up a proteomics core. And so have absolutely fantastic graduate students and colleagues. And just want to mention a couple of advertisements. So if you're interested in venoms, we put out the second edition of the book a couple of years back now. And next summer, there's a Gordon Research Conference that I'm a co-chair of. So make your travel plans now. And then I'd like to thank you for your attention. And if I have any time, I'd be happy to try to answer any questions you might have. Thank you.
All right, questions for Steve?
Yeah, super quick talk. I have a ton of questions, but I won't hog. Okay, so it's a single evolutionary event that created this branch. Question.
Yeah, I mean, there's a certain subset of people that believe that pushes it back way deeper in the clay.
The thing that's confusing is there's like this evolutionary genomic propensity to do this crazy elaboration and duplication, but they aren't related. I mean, there's a diversity of different toxins that have been generated from that. And so the comment about what were they called, microchromosomes? Maybe could that be the genomic driver that has allowed for this elaboration?
In part, I think that might provide more flexibility. And Todd's lab in particular has been looking at chromatin exposure experiments and how all that stuff is expressed. There seems to be steric interference and hindrance of too much chromatin exposed in the same small space. You can't fit all the machinery in there. So there's some weird things going on with that end of things, but these very different lineages, like the Gila monster and then the front fang venomous snakes, sharing some of the same kinds of components, those almost have to be convergent. You think, well, that's kind of bizarre because they're about as far apart as squamate reptiles can be. Not quite, but almost. But the other constraint that comes into that is that not everything is good as a toxin. To do a cell X or a protein extract from somebody and inject it to somebody else, you could probably kill them with that. It's not very effective and efficient. So what seems to have happened over evolutionary time is that the number of contenders has went down to a very small subset associated with secretory cells as well. And so that constraint on what's available to a secretory cell. So to me, it's not that surprising that we see the same kind of molecules popping up, the phospholipases, serine proteinases, metalloproteinases. And some of them are very stable and some of them are unstable, like the metalloproteinases are more labile, more problem to work with too. The three-finger toxins, I didn't really get into it as much as I could, but those are crazy. And they have activities, three-finger proteins, that basically all look like three fingers pointing down like that, stabilized by a cysteine knot up here. they're as divergent activities as alpha neurotoxins, very, very potent neurotoxins. Cholinesterase inhibitors, muscarinic receptor inhibitors. And then there's a couple of oddballs that are associated with salamander reproductive behavior and limb and tail regeneration in salamanders. So it's been talked about as this scaffold that is incredibly stable, and just by changing the fingertips around, you can engineer all sorts of different things. And a group in France actually did. They actually made an HIV metalloproteinase inhibitor from a 3-finger toxin that they just re-engineered some of that variable portion of the. So again, I think part of it has to do with the fact that of all those thousands of protein families possible, there's only a few candidates that really fill the bill. So.
Sorry. Yeah, thanks, Dave. That was great. I was intrigued by a comment you made in passing in your setup about the geographic variation in one of them and in the abundance of one of the peptides or something.
Yeah.
That from north to south would change quite a bit. Yeah, is that, am I interpreting that correct? Yeah.
No,
I was just interested in any follow-up on the microevolutionary patterns of, say, regulatory regions associated with that. So if there's abundance variation, you could explore upstream regions where there might be some relaxed selection of the mutation level.
I think that's probably going to be coming out in the next couple of years. But NSF just gave Todd and us a big pile of money. So it's asking exactly those kind of questions. We've looked at kind of the broad picture and what kind of transcription factors and such promoters are really being kicked up in high gear, and it's a very elaborate list. But getting back to that particular study, Kara just published that this summer. And so it's a very fascinating system where I've talked to a couple of you about it, that if you know where Interstate 70 goes across Colorado, that's basically our marker. That northern phenotype is found from there all the way to Canada. The southern phenotype is found on the other side all the way down. It has nothing to do with the freeway, but it's just a good visual marker for where that happens. And exactly why that's happening, we don't know. But the difference at the transcriptome and the proteome level, and the protein chemistry level, and the functionality level is just phenomenal. And it's just disjunct change right there. If you look at possibility of genetic isolation or differentiation in populations, coming up to that, it's just continuous. They don't seem to be, there's no trend that follows any north-south axis or anything like that. So it's a very curious scenario. We've tried to figure out, Kara tried to come up with an explanation for what this is. And there are a bunch of abiotic parameters that she modeled and suggested that temperature is one of them. We have dominant rainfall patterns or something like that was another that correlated with that, but it wasn't a real strong relationship. So yeah, so it wasn't like everything, answer part of one question, you have 15 others. So, right. Yeah.
If you looked at it developmentally, just like the different, like, you have this phenotype versus like Northern versus Southern, because I know like certain species of fish, just like abiotic factors can influence like the developmental biology to a point where like different structures are made at different like rates, and it just phenotypically differs from the adult version.
Well, actually, when I first started graduate school, what interested me was venom ontogeny, how it looked as a newborn to an adult. And so that we've done a couple of reiterations of that with very different species over the years. We've got that, but as far as the developmental mechanisms regulating that, it's still a little up in the air. And we have a colleague in Leiden that was very interested in doing that. Required me C-sectioning rattlesnakes on a certain basis. We tried to do that actually successfully, got them out, kept the mother alive and everything. Not only that, but I ended up turning her loose, recaptured her a year later. So my surgical skills are very. So anyhow, we don't really know on that end of things. We do know that it occurs. And there are changes that go this way, changes that go that way. So,
Let me just jump in here, so, because I know we're past there. Is there anyone on the online? No. All right, maybe one last question?
You got a question?
Yeah, if they could upregulate disintegrate, it would mean they wouldn't have to throw this way as much presumably somewhat costly
Yeah,
venom.
it seems
Why don't they do
Why
that?
don't they just kick that up?
Yeah.
That's a good question. And if you look at a variety of species, you still see all of them are fairly low like that. Part of that is that that's not the only reason why they're kicking up the level of venom. I mean, it's a guy at Loma Linda University that's looked at dosing. And if you look across the several species he's worked with, what you see, again, you compare the amount that they deliver to what it takes to kill even some of the stuff that we did with lizards, a native prey species. The overdosing is usually 2 to 4 orders of magnitude above those levels of toxicity. So they're really doing the job and then some. And the whole aspect of it being expensive to produce, it's probably not a whole lot more expensive than, muscle tissue or other body tissues. But if you use all your venom, then you don't have any left to defend yourself or next feeding bout or something like that. So probably the more important question from that end of things is not the cost per se, but instead whether or not you deplete what you have. So that tends to limit how much a snake typically injects to prey. So I don't know if that addresses your question, but
I still don't quite get why they don't
just upregulate.
basically, it doesn't matter. That's not a constraint, is what you're saying.
It doesn't seem to be.
Yeah.
Well, I'm just being whatever intrusive because I know we're past the hour. So, but we are going to be around here for this, right when we break up. So if anybody is dying to hear more about venom, so you can talk with Steve.
Yeah, I'd be happy to answer any
Right.
questions. I'm not going anywhere.
Yeah, let's thank him for